Hello, everyone, and welcome back to the 4th Annual Watershed Conference. My name is Sophie Glovier. I'm the Municipal Policy Specialist here at the Watershed Institute, and I am here today with Michelle de Blasio, and we will get started in just a few minutes. We're just going to give everybody some time to get on. Oops. Just a few logistics. If you have questions as we go through the program, please type them into the Q&A box. And I will ask Michelle when the time is right during the presentation, or we will just wait until we finish up. And again, if you're just joining us, we're just going to give it a minute or two for people to join, and then we'll get started. Okay, well, it looks like it is 2:30, so I am going to get started with introducing Michelle, and she is the Watershed Restoration Coordinator at the Nature Conservancy. And Michelle, while I'm introducing you, I will stop sharing my screen so you can share your slides. Michelle joined the Nature Conservancy's New Jersey chapter in 2015, and for the last 5 years has helped to lead the on-the-ground conservation efforts throughout New Jersey's northern freshwater systems. At TNC, she oversees the implementation of various freshwater conservation projects, ranging from floodplain restoration management, including planning and designing reforestation projects, to collecting, managing, and assessing water quality data for watershed scale monitoring. So we are very glad to have you here today with us, Michelle, to talk about gadgets, gizmos, and good science.
Hi, everyone. Thanks so much for having me join the conference this week. It still bums me out that we can't all get together in person, but we will make do with our virtual time together today. So I'm Michelle de Blasio. I'm the Watershed Restoration Coordinator, as you just heard. I've been involved in many different conservation projects with the Nature Conservancy since about 2015. And in some of my roles, what I'll talk to you today about is my particular role helping to oversee our water quality monitoring program that's happening in the Paul-and-Skill watershed. And as I mentioned, I've been involved in this program since 2015, which was when this program started, the water quality monitoring program started. And so I've had this unique opportunity to watch the program evolve over the years. And so I'm excited today to provide you all with what I like to say is a behind the scenes look into that program, and hopefully bring you all into the river with me to give you a better sense of how we collect scientific data to help tell the story of all our many different conservation projects happening on the ground. But before I begin, I wanted to just give an overview of the Nature Conservancy as an organization for those that may not be aware. So yes, we have a chapter here in New Jersey, but we are an international organization. We span across all 50 states and 69 different countries. Our mission is to conserve the lands and waters on which all life depends, and we have a vision to leave a sustainable world for future generations. And we've been around since 1951. In New Jersey, 1955 is when we got started here in the state. And we've been working on a number of different conservation strategies. Initially, our organization started as a land preservation organization, but as the landscape changed and the work changes, so too do our conservation strategies. So over recent decades, we've really evolved into these bigger, broader conservation strategies to really help tackle some of these big world problems that are happening on the ground. And so you can see here some of our conservation strategies that we're working on in New Jersey. I'll just give you a glimpse into what some of the on the ground work looks like to help us achieve these, embedded within these different strategies. So conserving resilient lands and waters. Our projects range from working to protect, hundreds of acres of intact forests that are needed for different state and threatened wildlife species like bobcats. We're also working towards increasing aquatic connectivity across the state via dam removals, improving water quality in certain areas. We do that through different riparian or floodplain reforestation projects. We're also very much involved working along our coast and working on different policies in terms of coastal restoration and really incorporating nature-based solutions into some of that work. So for instance, using different dredge materials to help build up some of degraded marshes. We have a new urban program, which we're really excited about. It's this greening our cities strategy, which is really working to incorporate nature-based solutions into urban and city planning and different ordinances. And last, but certainly not least, we have our Tackling Climate Change Initiative or strategy. And embedded within this, we have different projects going on involved with influencing offshore wind development in New Jersey to minimize ecological impacts. We're also working with partners and different companies to incentivize and advance key climate policies. So, you can see we work across the state. We have many different strategies. We have our work cut out for us, a really big agenda. But something that I think is always most interesting is all these different strategies and all the different work we do on the ground. It always has this overarching thread, if you will, that ties us all together, which is our water resources. And so everything we do on the land and in the landscape ultimately affects our water resources, whether that be coastal or freshwater. And so I want to give you just a kind of brief overview or background of our freshwater resources here in New Jersey and what that looks like. And it's a sad and grim reality, but about 85% of our rivers and streams here in this state are polluted. And there are, many different sources of pollution. So I'll just go through a few of those. We have our, point source pollutions, which stems from municipal or industrial wastewater discharges, plant discharges. We also see residential and urban stormwater runoff issues. We have our non-point sources of pollution. This is, anything from fertilizer that goes on people's lawns to make their grass greener or on farm fields. We have different, sediments that can pollute our water from, say, maybe a construction site or different croplands. Salt is actually a pollutant that harms our aquatic species in our streams. We see this from either irrigation or de-icing in the wintertime, bacteria from different, animal waste or wildlife waste. And so, there's many different ways that our waters end up polluted. And, when we start to think about how that is, thinking of climate change, you can think of, all these different things are impairing our waterways. And in the face of, climate change, these pollutants can enter our stream at faster rates. So they can start to, these impairments can be seen at different levels. So when we start to think about, as air temperatures begin to get warm, that's going to ultimately affect our stream temperatures for the aquatic species. When we start to see more severe storms, that's going to have an impact on flooding. It can have an impact and potential for failing infrastructure or in-stream infrastructure like dams. Increased stormwater runoff during these storm events. So all those different pollutants that can run off into our streams, those will just be exacerbated in time as the effects of climate change become more real. Sea level rise is another big concern. So as our sea levels rise, think about those brackish water systems like we see in the Delaware Bay, where we have salty water, and becomes a little bit more freshwater as you move upstream. Well, as those sea levels begin to rise and the saltwater begins to we call it saltwater intrusion. So you have this brackish system, but now it becomes more brackish as you move upstream. So that can have, really big impacts to the aquatic species that live in these systems, but also to our own drinking sources of water. The Delaware River, for instance, provides drinking water to millions and millions of people in the Philadelphia area. And so, when we think about water quality and climate change, it's, a lot to do with us and, the quality of our water that we depend on, but also for all the critters that live in the stream as well. And so we have, a number of different things that affect our water quality. We have these, big issues that we're hoping to address. But I think what's most important is, we need to understand what our water quality is today, what our baseline conditions are, and then that'll help us predict what it will be in, say, the future. And so here at the Nature Conservancy, we've been working for several years in one distinct watershed to really improve our freshwater resources in one specific drainage area. And that drainage area is the Pollenskill. And I'll get into a little more detail in the next slide. But I did want to just provide an overview of all of our different projects that we're working on in this landscape, in this drainage area. And so we have really large-scale wetland restoration projects, which is shown down here in this photo, which is the hyperhumus wetland restoration project. This is actually at the Pollenskill Wildlife Management Area, and we're hoping to, restore this degraded area, add some more sinuosity back to these streams. We see lots of impairments coming. This is right in the headwaters. The site alone is over 1,000 acres. We have, we've just planted about 58,000 trees throughout the watershed, which is a really exciting milestone for us, but we're, hoping to improve water quality via tree plantings, bank stabilization, providing canopy cover to the streams. We have a number of dam removal projects that are, have happened or are currently underway, which will help us to, not only increase aquatic connectivity for migratory and resident fish species, but will also help to improve those water resources in these select stream reaches. And then last and certainly not least, we have a really big land preservation effort going on in our Bobcat Alley Land Preservation Corridor, which is an area that connects the Kittatinny Ridge Mountains to the highlands, and it serves as an intact forest wildlife corridor for our state threatened bobcat. And so just to bring you kind of on the ground and give you a better sense of where all these projects fall within the Pollenskill, you can see this is the boundary, and the Pollenskill is the third largest tributary to the Delaware, which is this brighter blue. And as we start in the headwaters in Newton, which is in Sussex County, and the lower end is in Warren County, we have our hyperhumus project that I just described at the Pollenskill watershed, or I'm sorry, wildlife management area. And as the river drains north and then to the west, we hit this really large reforestation corridor. It's about 4 miles that we've connected with our tree planting projects. There's about 15 to 20,000 trees that we've planted in just this corridor alone. But you'll see throughout the entire watershed, all the other orange blobs also represent where those additional tree plantings are to hit that 58,000 that I mentioned earlier. And as you move downstream towards the Delaware, we begin to see all of our dam removal projects. And so we were successful in removing the Columbia Dam back in 2019. Which was the largest dam in New Jersey and ranked in the top 5% as a priority dam for removal in terms of increasing aquatic connectivity for migratory and resident fish species. And then as you move back upstream here, we have the Paulina Dam and the County Line Dam, both of which are slated for removal in the coming years. And so with these 3 different removals alone, we're hoping to reconnect roughly 45 miles of stream for these aquatic species along the main stem, but also within these tributaries. And then this dark green area with the darker green parcels identified, this is that Bobcat Alley corridor, that Bobcat Alley Wildlife Corridor that we are working to protect and preserve intact forests that is needed for bobcats and other terrestrial wildlife. But what we'll talk about today is all the orange dots that you see on the map. And those are all of our different water quality monitoring stations. And you can see we have them situated upstream, within, and downstream, some of our large project areas, upstream and downstream of our dams. And then we have some that are, scattered about throughout the watershed. And those are what we call maybe our integrative sites, or, just give us a better sense of a characterization of the water quality in the watershed with or without a project being associated with it. And so in order to capture changes to the water quality and ultimately project success, this monitoring program was really designed to provide adequate coverage to collect this, robust baseline data set and ultimately allow us to assess this pre-post project impact. And so there are a million different things you can measure when it comes to water quality. But again, we really wanted to make sure that what we were collecting was going to help to tell the story of our work and the success of our work, and to capture, the right baseline, conditions that we're hoping to see from these different projects. And so what science fit with our work and what information do we collect? We've identified a few key indicators that are really going to help to tell our story of these conservation projects. So I'll walk through a few of those now. So first up, we have our biological indicators of water quality. And this is simply the macroinvertebrates. There, you can see them on the screen. They're the critters that live under the rocks in the streams. They're a food source for many aquatic species, but they're most often used to assess stream health because, for one, they're relatively easy to collect and identify, but for two, each species differs in their tolerance to pollution, meaning that some species can survive in really very polluted waters, whereas others are super sensitive and they can't tolerate any sort of pollution. And so by assessing these macroinvertebrate communities, we're really able to not only get an indication of the biological health of that river system, but we can also indicate, it has other implications for what the quality of the water is as well, polluted, not polluted. So each spring, we hop into the river, we go to all those different yellow dots, all 23 of those sites. We collect bugs from those streams and then we send them out for analysis. And so this year, of course, COVID-19 definitely caused some delays in collecting from all these different sites. We were unable to hire any of our field staff that are typically out there in the streams collecting all these bugs. So, we had to reduce the number of sites, but we still got some data from our priority sites. And so we go in the stream, we collect the bugs. These surveys take about an hour or two to complete, because as soon as we're done collecting the bugs, we perform a habitat assessment. And so we follow the Environmental Protection Agency's rapid bioassessment for streams, which is simply a visual inspection of both the in-stream and the riparian area. And so each of these pictures here gives a good representation of what we're looking for. And so we're looking at different things in the riparian zone, bank stability, the amount of available in-stream cover, so something, and then it's rated on a scale of like 0 to 20 for each of these different physical indicators. And then at the end, you total it up and it gives you an overall rating of how excellent or poor the condition of the habitat is in. And so a photo over here, this would actually rank pretty high in that scale. For those habitat assessments, you can see you have areas of riffle run where there's probably a lot of oxygen there. You have a ton of available cover for both in-stream fish and macroinvertebrates. You have a nice forested vegetated bank on both sides. You wouldn't see too much erosion happening or excess sediment. So this would give you an overall rating of probably excellent and really high up there. Whereas if you look at something over here, this would probably give us somewhere in the middle to a little bit lower of a habitat rating. Although the in-stream habitat looks favorable to sustain life, and you have these riffle run areas and more free-flowing water systems, this riparian zone would probably drop your score down just a little bit. You see a parking lot, you see a mowed lawn with the tennis court, so there's not too much of a riparian area. And in addition to the rapid bioassessment, the visual habitat assessment, we also perform pebble count surveys. And rivers are dynamic systems. They're constantly flushing sediments through them. And so we use the pebble count surveys to really look deep into the riverbeds to assess the different sediments and the different composition of the bed materials. So, is it mostly cobble? Is it mostly boulders? Is it fine sediments, small particles? And so we take that information and then we assess the distribution of bed materials in these select survey reaches. And so a survey like this would become particularly important for, say, our dam removal projects where we expect drastic changes to those bed materials pre and post removal. And then we get into what I like to call more of our, hard science indicators. Again, there's a bunch of different things we can collect, but we've narrowed it down to, the top 4 physical chemical indicators of water quality that speak most directly to our projects. And so we'll go through just those pretty briefly. So temperature is a big one. This is something we're looking at. Temperature affects all living organisms in the stream. Certain aquatic species require certain temperature thresholds to survive and reproduce, like certain trout species can only reproduce within a certain threshold of temperature. So this is really important when we start to look at areas that have been deforested, like in our reforestation corridors, and when we start to, this is actually a photo from one of our oldest restoration sites, we can start to look at temperature where we're aiming to improve temperatures, or we're looking to decrease those in-stream temperatures through tree planting projects, where over time those trees will help to shade the river, provide that canopy cover that will ultimately bring those temperatures a little bit down. And then when you start to think about climate change and how that's going to affect it, it really gives us, it really has implications for how we do our work on the ground. So we can use this science, we can use this information to say, this is what the average stream temperatures are during the hottest times of the years. In these open canopy systems, we planted trees here with the tree cover. Now our streams are going to be at this degree, and we'll be able to meet standards for certain aquatic species. But then we can start to predict, if we start to see increases in air temperature, how will that affect our streams, water temperatures, and what kind of implications would that have for our on-the-ground work? Would we need to do and advance more work on the ground to make sure we can meet these temperature thresholds? So it's a very interesting, very futuristic thought, but it's something that we need to keep in mind and be aware of, and it's something that this data will really help to capture those influences in the future. So next up, dissolved oxygen. It's, again, it affects all species survival, just like we require oxygen, so do all the critters in the stream. Typically, cold, fast-flowing water is going to yield a higher concentration of oxygen in the water. And so areas like you see in this photo, where you don't really have any fast-flowing water, you're probably going to have really low levels of dissolved oxygen. Whereas something over here, where we have fast-flowing water, we're probably going to see a higher concentration. And so in areas like the hyperhumus project I talked about up in the headwaters, where we're trying to restore this very channelized, it's about a mile long along the main stem pollen skill, with no sinuosity, no riffles, no runs, where we're trying to bring that system back to a healthier stream reach. We would ultimately look to improve or increase those dissolved oxygen levels. So that's one thing we're looking at when it comes to dissolved oxygen. And ultimately, to meet those standards to, again, support the aquatic life. Connectivity. Connectivity is simply a measure of the water's capability to pass an electrical flow or electrical current. It's directly related to the concentration of ions in the water. Conductive ions come from, dissolved salts or inorganic materials such as sulfide, chloride. And so when you think about in the wintertime, when we salt our roads, and then we have a big storm or we have snow melt, ultimately that's going to end up, in our streams and kind of yield, you'll probably see a very high concentration of connectivity during those different periods. And so somewhere in Bobcat Alley, where we're, protecting these really large tracts of forest that surround our stream, we would expect to see really low and favorable levels of connectivity. And last, certainly not least, we have turbidity, which is simply a measure of the cloudiness in the water. So you can see here, usually this is increased during, storm events when the sediments are flushing through the system. But then sometimes we also see high levels of turbidity, not in any sort of storm event. And so areas like, say, above a dam or this, in-stream barrier where it's blocking sediments from free flowing downstream, we would expect to see really high levels of turbidity. And so this is a really important parameter to document changes to water quality when we start to look at our dam removal projects, because when those sediments are released over time, when they are released and settled out, we'd be able to bring those certain stream reaches back to more healthy levels of turbidity, rather no turbidity. You really don't want too much turbidity or too many sediments in your water. So that is, everything that we're looking at and how it kind of relates to our project. But I wanted to get into how we actually collect this information. And just like there's a ton of different things that you can collect, there's also a million different ways to collect this information. The macroinvertebrates and the habitat surveys and the pebble count surveys, those are pretty straightforward. They don't really require too much equipment. But these last parameters, the temperature, dissolved oxygen, turbidity, and conductivity, those require a little bit more, technical savvy to collect, if you will. And we use 3 different methods for that data collection, and we'll go through those. So first we have our discrete sampling, which discrete sampling is just using a handheld meter at one particular place at one particular time and taking a snapshot reading of what that is. And so there's a bunch of different types of meters that you can buy. You can see down here, this is a turbidity meter that measures just turbidity. In this photo, one of our seasonals is holding a YSI, which is actually capable of collecting connectivity, dissolved oxygen all on one meter. But again, there's a bunch of different meters you can buy that will give you this, snapshot reading of what that water quality is or what that parameter is. And so initially when we started this program, this was our main means of data collection, was going out to the stream X amount of times during the year, sticking our probes in the water and better understanding what the current conditions were. But the problem was that we were really only getting that condition at that particular time, and we couldn't really say anything about an average or really start to see a trend or a baseline into maximum values during the hottest time of the year. It required a lot of sampling, a lot of staff time, and it just really didn't yield the results we were looking for. And so again, to document these trends, we really needed to have a more robust data collection system to determine this baseline going forward. And so by year 2, we transitioned into this continuous in-stream monitoring methods, or CMI. And this is simply using, it's like the handheld meter, except it lives in the stream. And so we don't have to go out there all the time. We can put these in and set them at a certain time interval and come back out and offload the data. And so this allowed us to collect a ton more data and not spend as much staff time actually getting out there in the river. And so the 2 different meters that we've been using, you'll see here, it's the HL4 multi-parameter SON, which is seen here. They're a little bit larger, and this has all the 4 parameters that we're interested in on one sensor. And then we also use Onset's Hobo U26 dissolved oxygen logger, which collects temperature and dissolved oxygen. And just, loggers, SON, meter, they're all interchangeable. They basically just mean, a type of a piece of equipment that's collecting information. And then the different information is really based on the different type of sensor that you have at the end of that. And so we have 4 of those, of the hydro labs up here, situated throughout our Columbia Dam project site. And then we have 12 of these hobo loggers situated throughout the entire watershed. So they're strategically located at the select project sites and then throughout the watershed. But I wanted to give you a little bit of a better overview of how these actually work and how they operate. And so we don't just go out in the stream and throw them in the water and walk away. We actually strategically place them in the same location every single year. They actually get housed, we call it, inside this base unit. And so you can see it down here. One of our seasonals working to put the logger goes inside the base there. And so this base was actually a borrowed design from the folks at Delaware River Basin Commission. We assumed that, if they put their loggers in Delaware that experience super high floods and flows, that we felt pretty confident we could put our fancy equipment inside of these and put them in the Pollenskill, which doesn't even get close to the flows you see in the Delaware. And so it's a 60-pound, I always say nice and light, 60-pound bag of concrete that we pour the molds. We, drill out the PVC pipe, and then we construct it and put it all together. And then the logger just simply slides in. We cable wire it to the actual PVC pipe, and then we put a little name tag on it that, says, "Hi, I'm with the Nature Conservancy. I'm collecting, X, Y, and Z data. If you have any questions, give us a call, just in case, anybody's out there recreating and has any questions. They, know who this equipment belongs to." And so once they're in the bases and we go out in the stream, they're placed again, like I said, in the same reach, same location every year to ensure there's consistent, accurate data collection. And it's important that when you put these in the water, they're actually situated somewhere where it's not in direct sunlight and in areas where you experience a flow. So the water passes through the PVC pipe, passes through the sensor, just to make sure that everything's working properly. You don't want it to get clogged up. And so once they're placed in the river, they start recording data every 15 minutes. We place them in the river from about April through October. Again, due to COVID, we had to modify our sampling a little bit this year. We kept them out in the river for a shorter amount of time instead of that 6-month period. We just left them in for about 3 months to make sure we were capturing the hottest times of the year during those summer months, just to make sure we still had, we've had this baseline data being collected since 2016, so we didn't want to miss a year. We just didn't have the capacity to collect the full set. And once we're out there, once they're out in the river, we routinely check them. So we check them on a bi-weekly to monthly basis, and we do this just to make sure that there's, the loggers are there, there's no damage, that they're, where they need to be, especially if there's like a big storm or something, we would go out and check them. And sometimes we experience biofouling. So it's pretty much when different organisms kind of start growing on the sensors of the logger, which can actually give you poor readings or not accurate readings in the data. So we want to make sure that we clean them up. We want to cross-check them with a handheld meter to make sure that the calibrations are on point and you're collecting accurate, consistent data, and there's nothing going funky in the numbers. And so we analyze all the data when we get it back to the office for daily averages, maximum, and minimums, and then we look at those across the board. And so, when we switched to this continuous in-stream monitoring, we definitely were able to get a ton more data and didn't require us being out in the river every single day. But, it doesn't come without its challenges. And so although we can collect a lot of valuable information, we've definitely learned some hard lessons learned over the years. You can see here, sometimes we go out to check them and there's been a crazy flood that we can't even get in the river and check them. Or sometimes we go out and the water levels are so low that they're, somehow popped out of the water. Sometimes we go out and we find them on the banks of the river, which is always a questionable one. People recreate in these streams. We definitely, have had some issues, nothing too crazy over the years, but we just, it's good to get out there. It's good to check. These loggers are great. You can get a lot of valuable information from them. But as we began to think about more monitoring locations, as new projects began to come up in more recent years, we started to look into different means of data collection. So still continuous, but maybe new efficient means of collecting this information to avoid some of these things we were seeing with these, loggers that were sitting in the river over the years. So more recently, we transitioned to what's called remote sensing, which again is very similar to this. It still is collecting data, a rolling every minute or every 5 minutes or every 15 minutes. It's a continuous data set, except now instead of going out to the streams to offload that data and look at that data, you can go online and the data is being transmitted to an online portal, and then we can get that information live sitting on our couch. And so remote sensing is actually one of the most efficient means of data collection. But with that comes a pretty hefty price tag. And so some of these remote sensing stations and systems that you can buy through different vendors and stuff, they can cost like upwards of $8,000. So it gets pretty pricey. So although it's that's what you want, and that'll yield the most not cost effective, but the most effective means of data collection, you'll get a bunch. It might be hard for some people's budgets to really take these on. But fortunately for us, and a lot of our partners, we are part of this Delaware River Watershed Initiative, and we are in this, what we're calling Highlands Cluster. And basically what this initiative is it's a number of different organizations and entities working in all the different headwater streams that drain to the Delaware, implementing different conservation projects to improve the water resources in these headwater streams, which will ultimately improve the quality of the water in the Delaware. And so it's a really big, far-reaching program or initiative, rather, in that it's in New York, Pennsylvania, Delaware, and everybody's working together in these headwater streams. And so through this initiative, back in 2017, one of the partners that's a part of this, it's funded by the William Penn Foundation, and so it's been going on since about 2014, and a number of great things have come out. But in 2017, we had Stroudwater Research Center, which is one of the partners within this initiative, developed one of these sensor stations, and they, it was a mayfly data logger that one of their engineers had developed and started programming, and they started giving away these remote sensing stations to members of this initiative. And so, we took one, we put one up in the headwaters of the Paulinskill, and it was great. And the program became so big that they had to transition away from this, giveaway program to, essentially test this prototype throughout these headwater streams that drain to the Delaware. And then they started, kind of removing themselves. I mean, they used to come out, they'd put it in the stream for you. They'd, set everything up, and we were really more hands off. But in recent years, because the program's blown up so much, now they're transitioning to really make this more, in the hands of the users. And so this past year, we purchased, and they're also selling them, but they're selling them for like a way reduced price, anywhere from like $1,000 to $2,000, which is a steal in our eyes, because you cannot find these anywhere else for that sort of price. And so we're really excited to be able to obtain more of these sensor stations and get them at more of our locations. And so we just recently purchased 2 more of these, and we put them up and downstream of the Paulina Dam project. And when we got them, so Stroud used to put these together for us, but now, again, because the program got so big, they don't have the capacity to do that anymore. They're now training people how to put these mayfly sensor stations together, which at first, I'll be honest, I thought was going to be a little too technical, but their training was awesome and very thorough and very easy to follow. And essentially, what we did in the training is we learned how to program the software to speak to the sensors, which you can see down here. And then basically this logger, we programmed it to the software to speak to the computer and the online data portal. So this was the board. You put everything together and then it lives inside this box. It was a 2-day training that they had this May. Again, this was supposed to be an in-person training. Everything got a little hairy this year with COVID, unfortunately. But I will, just a big shout out to Stroud, because they did do an amazing job doing this, pretty technical training, but they made it very easy to follow. And so once we made these, turned everything on and got everything connected, put everything in the housing in the box, we go out to the stream and we position them into the stream bed. And so you can see here, the sensor is actually this red cable wire. The sensor where it collects the information is at the end, and they attach it to a PVC, and then the cable wire runs up along the bank, and this is where the actual logger is and where the data is being transmitted, and it sits on the side of the bank, and it's operated with this handy solar power panel. So it's, there's a backup battery in there, so it's, in case they don't get enough sun that one day. But we put these 2 sensor stations in, and once it's connected, it starts going live. So it goes immediately to this online platform that we'll go through in just a minute. But it's being transmitted by a cell signal, essentially. And so as long as you have cell coverage, you can get remote sensing data live at whatever interval you're setting it to. So these sensors, we have temperature, conductivity, and depth, and we're waiting on adding turbidity sensors to these as well. They're on a little bit of a back order, but we'll be able to easily go out and program and pop the turbidity sensors on. And these stay in the river all year. So the other continuous monitoring that we did, or that we do rather, that's only a 6-month period. These are actually able to stay in the water column for the entire winter, which is great because it's now opening up this entire year-round window that we never really experienced or had the opportunity to experience using just those continuous meters that sit in the stream. So how it all works, I wanted to give you just a few overviews of how this data sharing portal works. So when the data is being transmitted online, it's going to monitor my watershed, and it's a online platform that Stroud helped to develop. The link is right here, and this presentation will be shared. Anybody who is interested can go on and play around with it. But it's anybody, you don't need a login, you don't need a sensor station, anybody can log in at any time. You don't need a login name or credentials or anything like that. And when you log in, you'll see all the different sensor stations that are live and collecting data within the Delaware River Basin and beyond. So it wasn't necessarily exaggerating when I said the Stroud Water Research Center just didn't have the capacity, and this program just blew up. They've been able to provide these sensor stations to so many different partners and so many different organizations. There's over 450 stations where you see the green, those are all live and working. The grays are, almost up and running, and the reds are maybe the sensor's not working, but they're, planning to fix it. So when you zoom in, when you go to the website, this will be like the first page that you see, all the different stations across the basin. And when you begin to zoom in to your watershed or your site or an area of interest, it'll, you click on the green or the red or the gray, and it'll give you a little bit more information about that sensor. So where exactly it is, the coordinates, and the measurements. And then it has this, view data for this site button. You can search for sites as well. I just wanted to mention you can search by organization. So if you know of an organization that has a few sensor stations, you can just simply see, where all their sensor stations are situated along their river systems. You can search by a site code, or you can just use the map to, zoom in, zoom out to different areas of interest. When you click on the view data, it'll bring you to a big page that you see here. And it's going to show you all that data that's coming in. And this is a good visual for people that are operating to see, make sure that everything's working properly and it's collecting the information at the intervals. You can quickly see if there's a problem or there's something clogging your sensor, if these trend lines start to look a little different than what you normally see, every day when you log in and check your sensors. And so what I wanted to show you was how easy it is to take this data and easily interpret it and manage it and play around with the data. You don't really need too many skills in Excel to start to really just get a really quick understanding of what maybe the average is during a certain time of the year. So whatever parameter you're interested in, whatever sensor you have, so conductivity, again, I mentioned we have temperature and dissolved oxygen, but what you would do is you would click on this little arrow here, and that's going to bring you to this time series analyst, and you can go to your data sets or you can go to visualization. And if you go to the data sets, it's pretty much a matter of plug and play at that point. If you have two sensors like we do, you can put both those sensors on this same graph and you can compare, say, those upstream and downstream conditions at a dam site or any project site. If you wanted to look at how there's also an air temperature, a temperature for air on these sensor stations. So if you wanted, which is what's shown here, so if you wanted to look at, say, air temperature versus stream temperature, you can pull that up. You can pull it up on multiple loggers at one time. So it's really just a matter of clicks to get the information that you want. You can zoom into these certain time periods, you can zoom out, and then on the side it gives you all these different summary statistics. So this is actually a huge time save, because most of the data that we offload from those other loggers, we have to go back and there's software that helps to analyze different min, max, averages. But if you really want to start putting different loggers together and starting to make comparisons at different sites or up and downstream locations, it takes a little bit more time and effort to do that in the other software versus this online portal. And ultimately, this is, to me, the best way of collecting information, because you can look at the data anytime, anywhere. I can go on my phone and say, oh my gosh, there was a big storm last night. I wonder if anything's blocking that sensor. It really saves a lot of time in going out and checking your equipment that's sitting in these streams that are subject to, potential damage or down trees or anything like that. And so now what, we have, as I mentioned, 23 different sampling locations. We've got 16 of those continuous in-stream meters. We now have 3 sensor stations. We're looking to get more. We are, collecting as much information as we can in this watershed. I'd love to say we can just sit back and enjoy the show with these new remote sensing stations, but we still have, our work cut out for us, and we're in it for the long haul. I think that the most important takeaway from all this work is that it's going to really take decades, potentially, to see changes in water quality. And so it's important that the information we collect today, that we are collecting it, we're documenting it, we're seeing, what is the quality today? This is what we're doing on the landscape. These are maybe the potential impacts from climate change. And then this, is what we can assume or project it would be in years. But I think the bigger picture is, making sure we leave this information for our future scientists and for the folks that, come in 50 years and say, oh my gosh, look at how great this water quality is. I wonder what it used to be 50 years ago, or, so I think it's important that we're documenting this now. It's important to know what our freshwater resources are like. Are they impaired? Are they not impaired? Are they fixable? Can we, and I think it's, this program is really allowing us to do it in one distinct drainage area, which is really important. And we're excited to, continue to collect this data over the next 5 years and potentially beyond. But when all is said and done, we're looking at having about 10 years of baseline data. We are hoping to leave all of this data in the hands of the state. We don't want to hang on to this data. We want it to be accessible to anybody who's interested in making these water quality assessments of these streams and these tributaries in the pollen scale. And so, it's been a great program to be involved with, and I encourage all of you to get involved in maybe your own ways or in your own watersheds. Just like we have a pretty robust program in the pollen scale, you heard we have a ton of information to collect. We don't have a volunteer monitoring program just yet, because we've had our seasonals collect this scientific data for us. But there's potential in the future for us to begin a volunteer program, past our 10-year mark, but a bunch of other organizations are actually coordinating community science programs where they're getting out in the streams with volunteers. They've been doing it for years in certain watersheds like the Meskinekon and the Raritan. So I encourage all of you to check out your river, check out your stream, connect with your local watershed organization or entity, and see if there's a way to get involved if you're interested in going out in a day and seeing how this scientific data collection works, or if you just want to know simply what the quality of your water is in your own backyard. There's a bunch of different ways to get involved. Most notably, I'd say that the state is working really hard right now on this volunteer monitoring program where they're trying to better integrate all these different local watershed associations that are out collecting information and ensuring that all that data, just as I mentioned, the Nature Conservancy is making sure that our data lives in the hands of the state for future generations. So two is this volunteer monitoring program at the state. They're trying to better, like I said, integrate with these other nonprofit or watershed organizations to make sure they can get the data that is being collected through these volunteer programs. Excuse me. So there's a bunch of different ways to get involved. I always say, who doesn't want to get in the river? And so as long as it's nice, right? But I think, I encourage you all to also check out the Model My Watershed if you're interested in seeing any particular data set across the basin, or just want to learn more about average stream temperatures in your backyard. If there's a sensor station, you'd be able to, quickly find that out and check out some data, and there's contact information. So there's a ton of information out there. There's a bunch of people, great people out there collecting this information. And I would be happy to answer any more questions. I thank you so much all for your time today. It's always really fun to be able to present about this work and give you all like I said, a behind the scenes look into what it means to collect this science in these river systems. So thank you.
Well, Michelle, thank you. And there's a comment which I certainly agree with in the chat about what an excellent presentation this has been. So thank you for that. Just to go back to the beginning, when you started to talk about the indicators, temperature, turbidity, oxygen, what are the standards for these indicators? There's a question in the chat, and are they published somewhere?
Yeah, so the state has certain standards. So it's a great long story. So our state actually designates all of our streams based on different uses. So recreation, drinking, different like shellfish harvesting. And so depending on the designated use, is then associated with the surface water quality standard. So if it's used for this, then the standard for temperature is this. And so there's a ton of tables and graphs and different thresholds for each individual parameter. So yeah, so something like temperature, there are thresholds where if you want to sustain, looking at a designated use for recreation or for aquatic life, for, sustaining trout, your temperatures need to meet and not exceed a certain degree Celsius. And then it gets nice and complicated, and they give you, never to exceed this one standard, but then on a rolling average, it can't exceed this standard. So our state does have these standards set in place. And so that is actually a great question, because it's something that we use to base a lot of our goals on. And so most of our goals, water quality goals, are framed around, we want to have temperatures meeting the state standard in these certain stream reaches. And when you begin to work with the state and give them your data, they can start to use it in these statewide assessments of streams. And so if, say, we're finding in our data that temperature, for instance, isn't meeting surface water quality standards, then the state can potentially go in and do something about that in long short of it. But I can send or add a link to this presentation for the surface water quality standards in New Jersey. And again, it's turbidity, temperature, dissolved out, everything has its own standard. So I'd be happy to share that.
Great. And if you send it to me, Michelle, I can also send it to everybody in a follow-up email, everybody attending. There is also a comment that the sensor may need to be taken out during the winter if the stream tends to freeze over. So is that?
The sensor, It is true. So some of the sensors where some of our partners have in smaller systems or smaller streams, I definitely should have noted that as a warning. The pollen skill, the main stem, we don't experience that. But yes, in some of those smaller streams, if it freezes over, you do want to get that sensor out.
Great. And then I just had a question in the parts of your watershed where you've been working the longest and you showed those trees really starting to shade the water. Have you seen any increase in any of the in a positive way in any of the metrics that you're monitoring?
In the data, it's still too early to really see any clear indicator or sign that, oh, the trees are, decreasing temperatures. But I can say that we can finally see shade on the river, which is to us so exciting because, when you start to see it on the banks and you just know those trees are getting bigger, we will, that's the goal. We'll see it one day. So what we do for now is we have a whole other survivability survey that we do. And so we measure the success of the trees so we can assume if the trees are surviving, reproducing, then we'll be able to hit those water quality targets over time.
Got it. Well, I think that we have addressed all of the questions that we have. Anybody has a last question that they'd like to ask Michelle, please type it in now. Otherwise, I want to thank you, Michelle, for a really wonderful presentation. So we have a comment from Beth Styler-Berry. One quick response to the restoration was the return of the shad.
Thank you, Beth. It's like our highlight of our story. I think I was focused on our trees, not our dams. So yes.
Great. Well, that is a positive note to end on. And thank you so much for all the work that you do and for sharing this knowledge. And we'll be sure to get the links out to everybody with the follow-up email.
Thanks so much.
Okay. Goodbye, everyone.
Bye.